Welcome back to this next video in which we are discussing influenza. Okay, so we're currently in the process of discussing the pathogenesis of influenza, and we've just been discussing the replication cycle of the influenza virus. Now, fascinating though the replication cycle is, we can summarize it down to a very, very basic understanding, which is that the influenza virus comes into the cell. One influenza virus particle comes into one of these host cells, and thousands of genetically identical influenza virus particles leave that cell. And then the cell gets knackered by this and then ends up dying. So one virus particle comes into the cell, thousands of genetically identical or almost genetically identical influenza virus particles come out of the cell, and this destroys the cell that it's used, so the cell ends up dying. That is the simple way of summarizing the replication cycle of the influenza virus. Okay, now we have not finished the pathogenesis. That is not the end of our discussion of the pathogenesis. I have more things that I want to say. Firstly, I want to talk about how the influenza virus infection is going to spread down the respiratory tract. We've talked about it arriving in the nasal cavity. I want to talk about how it's going to spread down the respiratory tract. After that, what I want to do is talk about the inflammatory response that is going to occur to the influenza virus infection in the respiratory epithelium. Then what I want to discuss is how this inflammatory response is going to give rise to the local symptoms of influenza and also the systemic symptoms of influenza. And finally, to complete my discussion of the pathogenesis, I want to discuss the complication that can occur from having influenza, namely pneumonia. And of course, we've discussed the fact that rarely you can get primary influenza viral pneumonia. More commonly, you can get secondary bacterial pneumonia to influenza. So I want to discuss both of those. Okay, so the first thing then that I want to do is discuss how the influenza virus is going to spread down the respiratory tract. So we have discussed that we start off with a perfectly healthy person. This perfectly healthy person is then going to inhale loads of influenza virus particles which are going to come into the nasal cavity. What's then going to happen is most of those influenza virus particles will be destroyed by the mucus mechanism of it going back into the stomach. They'll get stuck in the mucus, and then the mucus will be moved gradually backwards, and they'll end up in the stomach and they'll be destroyed. However, if you inhale enough of the influenza virus particles, one of them will bypass this mechanism and get into an underlying ciliated columnar epithelial cell, and then the replication cycle will begin. That cell will start chucking out thousands of virus particles, which can then spread to the neighbouring ciliated columnar epithelial cells, and so the infection propagates. So, the nasal cavity is going to become infected by the influenza virus, and as I say, influenza viruses will spread from ciliated columnar epithelial cell to ciliated columnar epithelial cell, etc. Now, the viral infection does not just stay in the nasal cavity, however, it's going to move down the respiratory tract. And this makes complete sense, because remember, air is not stationary in the respiratory tract, it is continuously moving. When we breathe in, air is being moved down like this, and then when we breathe out, air is being moved up like this. So of course, in inspiration, you're going to be pulling air and any viral particles that end up in that air from the cells which are giving off viral particles, they're going to be brought down into the lower portions of the respiratory tract. So what's actually going to happen is the viral infection is going to move down the respiratory tract. It starts off up in the nasal cavity, it's going to then move back to the nasopharynx, the top portion of the nasopharynx, which remember is also covered by the same type of epithelium as the nasal cavity, this respiratory epithelium, the ciliated pseudostratified columnar epithelium, which is susceptible to influenza virus infection. So the infection is going to move from the nasal cavity back to the top portion of the nasopharynx. It can't then infect all of these bits here. So the next bit along that it can infect is the bottom portion of the larynx below the glottis, and indeed that's where it's going to spread next. So it'll go from the nasopharynx to the bottom portion of the larynx, then it spreads down the trachea and into the early bronchi. Now, I remember being quite surprised when I initially found this out. I didn't think that when you normally got flu that it would spread this low. However, it does. It does go down the trachea in most people who get flu, and it does end up in the early bronchi. Now, luckily, this spreading of the influenza virus down the respiratory tract takes time, and in that time, the body will be launching an adaptive immune response against the influenza viruses. And when that adaptive immune response kicks in, the influenza viruses are going to be annihilated from the respiratory system. So, this means that the virus does not continue to get further along the respiratory tract. If, however, you don't have a strong enough immune response, maybe you're immunosuppressed because you've got AIDS, or because you're taking immunosuppressants, or because you're on anti-cancer chemotherapy, or maybe because you're extremely old and the immune response does get weaker when you get older. For whatever reason, if the immune response, the adaptive immune response specifically, is weak, then the virus will be able to continue propagating down the respiratory tract further, and it will go into the smaller and smaller and smaller airways, and eventually it will get into the alveolar spaces, where it will be infecting the cells of the alveolar epithelium, the type 1 and type 2 pneumocytes. And the viral replication cycle inside those cells will be exactly the same as for the ciliated columnar epithelial cells. It'll be exactly like what we've just discussed, except you'd have to replace this picture that I've got here with one of the type 1 or type 2 pneumocytes. So, if the immune response was not there to eliminate the virus before it spreads that far, it would get down to the alveoli and you would then get primary viral pneumonia. Luckily, in most people who get flu, the immune response kicks in before it makes its way down that far, and it gets annihilated long before it ever reaches that stage. So in most people who get flu, the furthest it will get down is into these early bronchi, the great big airways. So, this means that influenza is both an upper respiratory tract infection and a lower respiratory tract infection. Because the nasal cavity and the nasopharynx are infected, it's an upper respiratory tract infection, and because the bottom portion of the larynx below the glottis and the trachea and the early bronchi can be infected, that makes it a lower respiratory tract infection as well. So, influenza causes both an upper and a lower respiratory tract infection. So that is how the influenza virus is going to spread down the respiratory tract then. The next topic that I want to discuss is the inflammatory response. So for this, we will find a clean portion of the board. We'll come right down here and we'll discuss the inflammatory response here. So before we go on to discuss the inflammatory response, let me just say a few words about the immune response, because we are overall going to now study the immune response, and this will take us a long time, and we're going to study the two different parts of the immune response. So remember, the immune system has two major arms, and these are the innate immune response, also called the inflammatory response. So the innate response, which is the inflammatory response, which is the one we're going to study now, and then also the second arm of the immune system is the adaptive immune response. So we will study this later on. Now, remember, the innate immune response is innate. It's a built-in mechanism. It's a set mechanism that you deploy no matter what the pathogen is. So if you get an infection in any tissue of your body, you deploy this same old response, which is the inflammatory response. And it's the same response no matter what pathogen you're dealing with. It's a non-specific response. It's not specific at all to the pathogen that you are dealing with. However, regardless of that, it is very effective. In some pathogen cases, it will completely annihilate the pathogen, not in the case of influenza usually. So the innate immune response, we'll study it. It occurs first. That's the great advantage to it. It occurs within minutes of the infection having occurred, whereas the adaptive immune response takes a long, long time, days to initiate. However, it is a specific immune response. It is specific to the type of pathogen that you are dealing with, and that's what makes it more powerful than the innate response. So they both have advantages. The innate immune response is extremely quick, whilst the adaptive immune response takes days to initiate, but the innate immune response, because it's not specific to pathogens, it's not as good at dealing with the pathogen as the adaptive immune response is. Now, because the innate response occurs first, we will be discussing the innate response first. So we'll discuss inflammation, and actually, you only need to discuss inflammation in order to justify where the local symptoms and the systemic symptoms of influenza actually come from. You do not need to know anything about the adaptive immune response to influenza in order to understand where the local and systemic symptoms of the disease actually come from. The local symptoms of influenza come because of the inflammation that's actually occurring in the respiratory tract. And the systemic symptoms come because when you have that inflammation occurring in the respiratory tract, loads of inflammatory mediators are released into the bloodstream, which will therefore lead to the systemic symptoms. So by studying inflammation, we are going to understand where both the local symptoms of influenza and the systemic symptoms of influenza actually come from. So let's begin then. Let's begin studying the inflammatory response. I'll underline inflammation, that's now our title. So, I'm going to begin by drawing the respiratory epithelium that has become infected by the influenza virus. And remember, in most cases of influenza, it's only respiratory epithelium that's going to be infected. The nasal cavity is infected, the nasopharynx is affected, bottom portion of the larynx, the trachea, and the early airways. They're all lined by respiratory epithelium. Remember also that the viral particles, when they're being released, when they're budding off from one of the ciliated columnar cells, they bud off on the apical side, not the basolateral side, which means the virus does not go deeper than the epithelium. That's why it does not cause a systemic infection. Okay, so it is just mainly going to be the ciliated columnar epithelial cells that are going to become infected. So I'm just going to draw a bit of the ciliated columnar epithelium out again. So here, in big thick red, is of course our basement membrane, and now on the surface of the basement membrane let's put some of our ciliated columnar epithelial cells. So we'll just draw a few of them, and I'll omit the other types of cells of the respiratory epithelium, so the basal cells and the goblet cells. I'm going to omit those. We'll just have the ciliated columnar epithelial cells, which are going to be the major type of cell that's going to be infected. So here are their cilia here. So we're discussing the inflammatory response. What is it that's going to actually cause the inflammatory response in the influenza infection? So let's say that this ciliated columnar epithelial cell here has become infected by an influenza virus. It's now churning out influenza virus particles, and this is going to kill that cell. So that cell is going to die. So I'm going to get some big dramatic colour here. We'll have big dramatic purple. It's going to die. So I'll put a great big cross for it. That cell has died now, let's say. And I should stress, influenza can do massive damage to your respiratory epithelia. Absolutely massive damage. You can lose a huge number of cells from the epithelium. The epithelium can become really, really bare when you get influenza. And the basal cells have then got a huge amount of work to do to repopulate the epithelium and reproduce all of the ciliated columnar epithelial cells that have been killed by the virus. So they are a huge victim to the influenza virus. A huge number of them are going to die when you get flu. Okay, and it is these cells dying that is going to cause the inflammatory response in the epithelium. So whenever a cell dies, it releases a huge number of molecules from its cytoplasm into the extracellular fluid, and a lot of these molecules are molecules that you would never ever normally find in the extracellular fluid. The classic example is ATP, adenosine triphosphate, the biological energy currency. This is a molecule that you find in a huge amount inside the cytoplasm of cells, but it's a molecule that you should never, ever, ever find in the extracellular fluid. So if you do find some ATP in the extracellular fluid, what would you interpret from that? If you were a little man walking around some extracellular fluid of your body, and you found some ATP. What would that mean? That would mean that somewhere nearby cells have died. And that would mean that there is probably a pathogen present in the tissue that has killed those cells. So, ATP is not the only molecule that would indicate that. There are a whole bunch of molecules that would never normally be found in the extracellular fluid, and if you do find them in the extracellular fluid, it is a giveaway sign that cells are dying and that there is a pathogen present in the tissue. And we have an umbrella term for these types of molecule that give away that there is a pathogen present inside the tissue and that the cells are dying. They are called DAMPs, for damage-associated molecular patterns. So this stands for damage, or some people actually call it danger. So D, either you can put that as damage or danger. So damage-associated molecular patterns. So they are molecules or molecular patterns that are associated with cellular damage. So it's a reasonably sensible name, damage-associated molecular patterns, and for short it's abbreviated down to DAMPs. So as I say, this is an umbrella term. This covers loads and loads of different molecules that all are of this basic principle that they should not be in the extracellular fluid, and if they are present in the extracellular fluid, it indicates that somewhere nearby cells have died, i.e. that damage has occurred. Now, these are the molecules then that are going to activate sentinel cells, the guard cells of the epithelium, to initiate the inflammatory response. So remember earlier on we discussed sentinel cells. These are guard cells that you have in any tissue in the body which are continuously on the lookout for signs that there is a pathogen present, and if they find signs that there is a pathogen present, they release loads of molecules called pro-inflammatory mediators that are calls for help and which are going to call for troops to come from the blood to help fight the pathogen that is present in that tissue. And therefore, we need to get those troops out of the blood, and that response where we move troops from the blood to the tissue, that is called the inflammatory response or the innate immune response. So let's do this in more detail then. So, next thing we need to do is add onto the picture then some sentinel cells. So I'll start by adding on a dendritic cell here. So remember, dendritic cells are one of the major types of sentinel cells that you have in all tissues, and they have these dendrites here. So this is a dendritic cell, or DC for short, one of the sentinel cells. Another major type of sentinel cell, which I'll have in this pink colour here, are the resident macrophages, and they'll be in the underlying connective tissue underneath the basement membrane here. So remember, they are a great big blob of a cell, and I will abbreviate them down to M5. Okay, so here we now have on our picture our two resident or sentinel cells here. So these damage-associated molecular patterns from the dying ciliated columnar epithelial cell are going to activate these sentinel cells to release the pro-inflammatory mediators. How are they going to do that? They're going to have receptors on the surface of the sentinel cells which they will bind to and activate. So I'll draw this here. So here, these little blobs are representing receptors on the surface of the sentinel cells that are going to be activated by these damage-associated molecular patterns. Now, remember, there are loads of different damage-associated molecular patterns, and all of them will have different receptors. So the sentinel cells are absolutely covered in receptors for different damage-associated molecular patterns. Okay, and there is again, an umbrella term to cover all the different types of these receptors for these molecules that give away the fact that there is a pathogen present, and these are pattern recognition receptors, or PRRs. So PRRs stands for pattern recognition receptors. So these are receptors that are capable of recognizing these patterns that indicate that there is a pathogen present. So pattern recognition receptors. So what will happen is DAMPs will bind to their pattern recognition receptors, and as I say, different DAMPs will have different pattern recognition receptors. These pattern recognition receptors will activate, they'll activate signaling pathways inside the sentinel cells that will then cause the sentinel cells to start producing and releasing pro-inflammatory mediators. So let me mark this on now. So these are going to start producing pro-inflammatory mediators, and they will go into the extracellular fluid. So they're going to release pro-inflammatory, meaning that they are in favour of the inflammatory response occurring, mediators. Now, there is another term that people often use instead of calling these pro-inflammatory mediators. However, pro-inflammatory mediators is the best term. You'll often hear people talk about pro-inflammatory cytokines when they're talking about the innate immune response. Now, let me tell you about what the definition of a cytokine is. A cytokine is a protein released by a cell of the immune system, which these sentinel cells are. Sentinel cells are cells of the immune system. So they're proteins that are released by cells of the immune system and which will go and bind to a receptor on another cell and change the behavior of that cell. So let me say that again. A cytokine is a protein released by a cell of the immune system which goes to another cell and changes the behavior of that other cell. Now, pro-inflammatory mediators are all molecules that are released by these cells of the immune system and which are going to go and change the behavior of other cells. However, they are not all proteins, and therefore they are not all cytokines. So a pro-inflammatory mediator is not necessarily a pro-inflammatory cytokine. All of the pro-inflammatory cytokines are pro-inflammatory mediators, but they're not all not all of the pro-inflammatory mediators are necessarily proteins, so they're not all necessarily cytokines. So be aware of what this term actually means, because there's a lot of confusion with what pro-inflammatory cytokine actually means. So let me give you then some examples of pro-inflammatory mediators. Now, there are absolutely tons of the things. I am not going to give you every single one. I am going to give you 5 of them. And these 5 are really, really important. We will start off with the most important 2, the ones that if you are going to learn any, you must learn these 2's names. Okay, so the 2 most important ones are interleukin-1, which is abbreviated to IL-1 for short. So this is an example of a pro-inflammatory mediator, a really important one. And it is in fact a pro-inflammatory cytokine, and it's a protein that is synthesized by the sentinel cells and released by them. And then the other really important example of a pro-inflammatory cytokine is, TNF it should be. TNF alpha. And this stands for tumor necrosis factor alpha. So the T is for tumor, the N is for necrosis, the F is for factor, and then it's tumor necrosis factor alpha, so TNF alpha. Okay, so both of these are proteins released by sentinel cells and which are going to trigger the inflammatory response. They are the most important ones. As I say, if you're only going to learn 2, you must know the names of those. They are absolutely key. Okay, now I'm going to give you 3 more that are slightly less important for medical students to know the names of than those 2, but which are nevertheless really important. So, third one, give me the pen. Third one is histamine. Now, histamine is certainly not a pro-inflammatory cytokine. It is not a protein. It is a very small molecule that is synthesized from the amino acid histidine. So, histidine is an amino acid. If you take the carboxylic acid group off that amino acid, you end up with histamine. So it's a small molecule. So we call it a pro-inflammatory mediator, but it's not a pro-inflammatory cytokine. So histamine is another very important one. Another pro-inflammatory cytokine now is interleukin-6. This is again a protein that is pro-inflammatory, and we'll discuss the function of interleukin-6. It's very important in triggering the systemic symptoms of inflammation, particularly in producing fever and things like that. And so that's why I want interleukin-6 to be on this list. And then another really important one, the one that's really important for producing pain, is prostaglandin E2, PGE2. And again, this is going to be important in the systemic symptoms as well. So prostaglandin, PG stands for prosta, and then glandin, and then this is specifically prostaglandin E2. There are multiple different prostaglandins. Prostaglandin E2 is not a protein, it's a lipid molecule, or at least it's derived from lipid molecules. So again, it's a reasonably small molecule compared to proteins. So those are the 5 pro-inflammatory mediators that I want you to know the names of. If you're only going to learn 2, these ones are the ones that you must know the names of, whereas these 3 are slightly less important, but still they're going to be important in my explanation of where all the different symptoms come of the influenza illness. Okay, right. So, these are the molecules that sentinel cells now release into the extracellular fluid, and they are going to cause inflammation. Particularly these 3 here, interleukin-1, tumor necrosis factor alpha, and histamine, they are going to cause the inflammatory response in the microvasculature. We'll talk about what these 2 do later on. So, these are cause for help, remember, from the sentinel cells, and what we are trying to do fundamentally is get troops that are all circulating in the bloodstream out of the bloodstream into the extracellular fluid near this epithelium. So we want them to come into the underlying connective tissue of the epithelium, and we want them even to come into the actual epithelial layer itself, and we want them to fight the pathogen that is present here. So, this is going to require some change in behaviour of the microvasculature blood vessels. So before we can actually discuss the inflammatory response, we need to have a quick revision of the structure of microvasculature blood vessels. So I'm just picking a colour to do this in. We'll do it in the strange purple colour. So, a bit of a revision now of the microvasculature blood vessels. So, microvasculature refers to tiny little blood vessels of the circulatory system. And these are the sort of blood vessels that we're going to have in the underlying connective tissue here that are going to respond to all of these pro-inflammatory mediators that have been released by the sentinel cells. And that's why I want to do this little bit of revision of the microvasculature. So, firstly, a very basic picture. The microvasculature consists of the terminal arterioles, which then split into capillaries, which then reconverge into post-capillary venules. So let me just draw a basic picture of this. So this can represent the terminal arteriole. Then here we're going to have the capillary bed. So here the terminal arteriole has split into a capillary bed there, and then they reconverge up to form the post-capillary venule. So let me just label these up. And these are really tiny little blood vessels. They're the tiny little blood vessels that you have in a local little portion of tissue. And as I say, in this underlying connective tissue here, you're going to have these tiny little blood vessels. So on this side, we have the terminal arteriole. Now remember, the word arteriole covers a huge range of different sized blood vessels. It's a really bad word, actually, because it covers blood vessels that are just smaller than arteries, right the way down to these tiny little blood vessels, the terminal arterioles, which are the blood vessels just before the capillaries. So to emphasize the fact that I actually mean this arteriole just before it has broken up into capillaries, we put the terminal in front of there. So you should not just put arteriole, because arteriole actually covers a whole scope of different sized blood vessels, some of which are only just smaller than arteries. So we have the terminal arteriole, which then breaks down into the capillaries here. So they're the ones in the middle, and then we get the capillaries reconverging up into a venule here, but again, venule is a term that covers a huge scope of different sized blood vessels. So to emphasize the fact that we mean a venule just after the capillaries, we put post-capillary in front. So a post-capillary venule is a venule just after the capillary bed, where the capillaries have just rejoined together, and then you've got this tiny little blood vessel that is only barely bigger than capillaries, called a post-capillary venule. So these are the 3 components of the microvasculature. The terminal arteriole here, the capillaries of the capillary bed here, and then the post-capillary venule after the capillaries. Okay, now I want to do a little bit more than this. I do want to just go over the structure of these blood vessels. So for each one, I want to take a cross section through and show you the structure of these so that we totally understand the size of these blood vessels and the wall structure of these blood vessels. So let's start off with the most complicated one, which is the terminal arteriole. So again, the terminal arteriole is going to be lined by endothelium, which is a type of epithelium, and these endothelial cells of the endothelium will be sat on a basement membrane. So I will start by drawing a little basement membrane here, and again it's in the bright red colour. And then once I've got the thinner pen back, we'll now draw some of the endothelial cells. So these will be sat on the basement membrane, and again remember, basement membrane is this meshwork of proteins and carbohydrate fibers to which the epithelial cells, or the endothelial cells in this case, are attached, and this is the reason the endothelial cells don't just fall off into the lumen. So here are some endothelial cells facing into the lumen of the blood vessel, and I hope this sort of gets across the size of this blood vessel. These are single cells, so the diameter of this blood vessel is not that big. You could fit a few red blood cells across the diameter of this tiny terminal arteriole. It's a tiny little blood vessel that will be in the connective tissue underlying the respiratory epithelium. Now, you have now other things around the outside of the basement membrane. You're going to have smooth muscle cells. So I'll go to purple now. Around the basement membrane, you're now going to have a layer of vascular smooth muscle cells, which I'm now colouring in here in purple. And I realise I haven't actually put any labels on this picture. So those cells in white, those are endothelial cells that line the inside of blood vessels. Then, of course, in red, we are very familiar with the basement membrane. And then surrounding the basement membrane, you're going to actually have smooth muscle cells. So let me draw one of these in. So these are spindle-shaped cells. They have a thicker body and then narrow edges. So I'll draw them in here. Here's another one. And they are connected together in these sort of like rings of smooth muscle cells, like so, as I'm showing here. And you'll have multiple different layers of these. It's not just a single layer of these cells, but they form these sort of rings of smooth muscle cells like this. Okay, and the name for these is vascular smooth muscle cells, which for short can be abbreviated down to VSMCs, vascular smooth muscle cells. Now, the significance of the fact that these vascular smooth muscle cells form these rings is that imagine what would happen if every single one of these now starts contracting. So if this contracts, this contracts, this contracts, this contracts, this contracts, they all shorten their length, and that means that the diameter, the circumference of these rings will decrease. And if the circumference of a ring decreases, now it's time for the diameter of the ring will decrease. And therefore the diameter of the lumen of the blood vessel will also decrease. So the important thing that I hope you've gained from this picture is the fact that the diameter of terminal arterioles is controlled by the vascular smooth muscle cell layer that surrounds or is part of the wall of these terminal arterioles. So that's the terminal arteriole. Let's now do the capillaries. Now capillaries are absolutely tiny. So let's do it. So again, back to vivid red for basement membranes. Capillary, I'll try and put it in scale to the terminal arteriole. So it's a tiny little blood vessel. So there's its basement membrane, and literally a single endothelial cell can cover can go the entire way around. So I'll put a single endothelial cell, and there can be its nucleus. So it is a tiny, tiny little blood vessel. This is our capillary. And you can see that in the lumen, you could just about fit a single red blood cell through that lumen. So they are tiny, tiny little blood vessels that will absolutely, most definitely be in this connective tissue. So the terminal arterioles break up into lots of little capillaries, which form the capillary bed. And then what's going to happen is the capillaries are going to reconverge up into the postcapillary venule. And by the way, this is it. That's the wall structure of a capillary, just the endothelial cell on the basement membrane. And remember, the reason for that is that the capillaries are where gas exchange occurs. Oxygen will leave the blood and go into the tissue, and carbon dioxide will come from the tissue back into the blood, unless of course we're talking about pulmonary capillaries, the ones underneath the alveoli, where it's the opposite way round. Oxygen comes in, carbon dioxide goes out. If we're talking about a capillary at some peripheral tissue, oxygen leaves, carbon dioxide comes in. Okay, so you want the wall to be as thin as possible so that gas exchange is maximised. So that is our capillary. Now let's do the post-capillary venule. So back to the vivid red and the thick pen. So we'll do our post-capillary venule up here. So here is the basement membrane of the post-capillary venule. And then let's put the endothelial cells now on. So like so. So here's one endothelial cell. Here's another endothelial cell, another one, another one, another one. So as you can see, it's a much bigger blood vessel than the capillaries, but that's the wall structure finished. That's my picture of the post-capillary venule finished. There are no more layers, it's just the endothelium sitting on the basement membrane like so. So it's a very, very thin-walled vessel. It's like a giant capillary. So the capillaries all join back together to form this post-capillary venule. And this is the structure of the microvasculature. You have these types of blood vessels in every tissue of the body. You have terminal arterioles breaking down into capillaries and then reconverging up into post-capillary venules, and they will be present in this connective tissue underlying the respiratory epithelium, and they are the vessels that are going to respond to these pro-inflammatory mediators that have been released by these sentinel cells and are actually going to initiate the inflammatory response. Now, I'm going to take a break here. In the next video, we will actually talk about the inflammatory response that is going to be initiated by these pro-inflammatory mediators.